Hello and welcome to the Student Disease presentation of 2021. My name is Michael Shen and I'm joined today by my partner,
Rasheed.
Today we'll be discussing the Ebola virus.
To begin with, the Ebola virus, EBOV, which was named after the Ebola River, was first discovered in 1976 following various outbreaks within Sudan and Congo, eventually leading to the establishment of 5 different species of the virus. Ebola virus causes Ebola virus disease, EBD, formerly known as Ebola hemorrhagic fever.
The 5 distinct subtypes of Ebola have been named as follows: Zaire, Bundibugyo, Sudan, Reston, and Thai forest, the deadliest strain being Zaire with a 90% mortality rate. The average case fatality rate of Ebola overall is 50%. Overall, EBOV is a resilient virus as it takes a high level of gamma radiation, UV exposure, and long periods of heat, 60 degrees Celsius, to kill its molecules.
In terms of epidemiology, since the discovery in 1976, there have been 20 documented outbreaks. Most endemic outbreaks of Zaire and Sudan Ebola virus are within rural areas of Africa from consumption of contaminated meat. The most recent outbreak documented was in June of 2020 in the Democratic Republic of Congo. The longest outbreak and epidemic affected the United States, Europe, and areas of Western Africa in 2014 and caused the initiative to look into this virus regarding vaccination development.
The Ebola virus belongs to the Filoviridae family, a name derived from Latin, in which phylum signifies thread, and is a filamentous virus with a thread structure. It is important to note that Filoviridae, from the Mononegrivalarius order, are negative single-stranded RNA viruses with heterogeneous structure.
Ebola virus cells contain a cell membrane with nuclear proteins inside itself with an outer glycoprotein layer. Its genetic material, RNA, is within the nuclear protein. It also contains polymerase, which aids in replication. The VP40 and VP30 proteins essentially help in bioreplication as well.
The Ebola virus consists of 7 genes that encode for at least 10 different known proteins. These proteins are involved with encapsulation, polymerase cofactor, synthesis of viral RNA, nuclear protein regulations, as well as antibody and interferon defense. Ebola virus infection is a complex process in which the mechanism is not fully understood. However, from the overall analysis, there is a general idea. Attachment occurs through the glycoprotein 1 subunit of EBOV, which has a receptor binding domain which interacts with multiple cell receptors, a glycan cap which protects binding insites from cellular antibodies, and interacts with glycoprotein 2 for internal fusion regulation, and a mucin-like domain which protects from immune recognition. Ebola virus uptake by the cell upon attachment occurs through different ways: micropinocytosis, glycoprotein-mediated fusion, as well as clathrin-mediated endocytosis. The uncoating and fusion occurs through GP2 and GP1 glycan cap interaction for fusion regulation. The fusion loop has 16 amino acid hydrophobic sequences which insert into the host membrane to begin fusion. Unknown enzymes then trigger and accelerate the fusion process. Fusion loop of GP2 is then inserted into the host membrane, where heptad repeats of the GP2 create a bundle that opens the membrane for RNA and associated proteins to be released into the host cytoplasm for replication. A ribonucleoprotein complex is then formed with nucleoprotein encapsulated EBOV, ssRNA, and RNA polymerase, in which the RNP serves as a template for the production of cRNA, which generates virogenomic RNA packaged to virons. Within the perinuclear regions, the nucleocapsids are formed and transported to budding sites at the plasma membrane.
In terms of transmission, the 2 main routes are animal to human and human to human contact. The first infections in humans are perhaps caused by the consumption of meat of non-human infected primates. Human to human transmission is the main route of transfer. The most infectious bodily fluids are blood, semen, urine, saliva, feces, sweat, and vomitus. While transmission has been detected in breast milk, breastfeeding is still not considered a confirmed route. Ebola virus disease can be classified as zoonotic. Humans are accidental hosts as they can become infected through contaminated meat or close contact with animals such as monkeys, gorillas, and bats. It has been confirmed that Ebola cannot be contracted through air, water, or food, but only from contact with the bodily fluids of an infected organism. Ebola virus infection is generally characterized by high inflammatory cytokines, weakened immune response, septic shock, coagulation disorders, and multi-organ failure from high hemorrhagic fever. Initially, diagnosis of Ebola is challenging, as its appearance usually overlaps with multiple differential diagnoses, such as malaria, dengue fever, typhoid fever, and meningococemia. Presentation of symptoms starts after an incubation period of about 3 to 21 days. The disease infection itself lasts about 4 to 15 days. Symptoms typically develop abruptly in 3 phases: fever, chills, inflammation, headache, myalgia, general malaise, usually then followed within the first week by progressive GI symptoms such as anorexia, nausea, and abdominal discomfort. Following infection, the initial incubation period is non-contagious, allowing the appearance of viral symptoms to indicate the beginning of contagiousness.
The overall immunosuppression and uncontrolled inflammation are causes of pathogenesis. Infected monocytes and macrophages cause oversecretion of pro-inflammatory mediators, along with reduced adaptive immune response from T cells. Survivors, however, display immense and long-lasting CD8 T cells. As such, rapid induction of antibodies and CD8 T cells in immune response may be used for protective immunity.
The risk of death can occur from as early as 6 days of onset symptoms. The inflammatory molecules released by the immune cells can cause damage to vascular integrity, leading to hemorrhagic fever. The coagulation cascade is disrupted due to diminished platelet aggregation from the release of cytokines. This is typically the cause of death following EBD infection, as the resultant cell damage leads to the potential development of heart, liver, lung, and/or kidney damage. It's important to note that this does not always occur, and it is in fact possible to survive the illness. In terms of the aftermath of Ebola virus, survivors will develop antibodies. Several cases have later reported associations with hepatitis, uveitis, and spinal cord injury in the long run. An additional issue seen in survivors has been cell damage due to the virus's release of cytokines. Currently, the only reliable preventative measures for EBD are isolation of infected persons and the use of infection control material. Specific examples would include case management, detailed surveillance and contact tracing, and a reliable laboratory service. While there is no licensed treatment, early supportive care involving rehydration and symptomatic treatment can ultimately improve survival.
In 2018, 36 vaccine trials were completed, with 14 ongoing, yet no approval of vaccine use or administration has occurred. The Ebola candidate vaccines use recombinant vectors based on human adenovirus serotypes, as well as chimpanzee adenovirus serotypes, expressions of the Zaire Ebola virus strain. The VP30 regulatory protein is of target due to its role in transcription and translation, as well as the glycoprotein due to its role in attachment, fusion, entry, and immune invasion. Furthermore, there has been other research regarding the virus's effect on the skin microbiome, suggesting that microRNA of the virus, small regulatory non-coding RNA, play a role in regulation of endothelial cell rupture and immune escape of host defense mechanisms, as the virus is found to spread through direct skin contact. Research has looked into the skin microbiota, such as the species Propionobacterium acnes, and its role as a skin immune shield to interfere with viral microRNA through regulation of expression of a common thrombospondin protein as a target gene, which plays a role in initiating cell-mediated immune responses in the skin.
Here are following references used in this presentation. Thank you everyone very much for your attention. We hope you learned something new today regarding Ebola virus disease.
Please let us know if you have any questions. Thank you.